To identify functionally relevant SNPs, we applied the SNP-F annotation tool to the chromosome 22 variants. SNP-F describes the type of variant, affected transcript type, and the putative impact. For downstream analysis, we focused on 16 variants across 10 genes with the most risky high and moderate impact mutations in protein coding transcripts specifically to highlight protein structure changes. High impact SNPs are stop gains and splice donor variants, while moderate are missense mutations.
After selecting 16 variants for further analysis, we used AlphaFold to generate structural models of the reference protein in blue and mutant proteins in pink for a total of 32 structures. Analyzing them revealed that a majority of the mutations were found in alpha helices, and a majority of the mutations likely have minimal effects due to relatively undisturbed biochemical properties. Viewing the models for APOL4 here as a representative example, it should be noted that AlphaFold struggled to place protein domains, though typically had a higher confidence overall for the reference structure when compared to the mutant. Additionally, AlphaFold struggled to predict the structural impact of varying mutations. Proline is known to kink alpha helices. AlphaFold correctly predicts this kink when proline is in the native structure, and correctly removes the kink when proline is mutated away. However, when proline is non-native in an alpha helix and then mutates into an alpha helix, AlphaFold misses the kink and does not predict it. Thus, we mainly relied on structural and biochemical knowledge to predict the impact of each mutation.
We took a closer look at the APOL4 gene and the variant that likely has a removed kink within one of the largest alpha helices in the protein. We know that AlphaFold isn't perfect, especially for comparing the structure of a sequence with itself after a point mutation in the protein sequence. However, regardless, we did a molecular dynamics simulation for 20 nanoseconds to see how well the normal protein and mutated protein would bind to a membrane. Due to limited compute, we only ran it for 20 nanoseconds and looked at the non-bonded interaction energy between the protein and membrane at the end of the simulation as a proxy for binding affinity. We found that the normal protein with the kink more easily binds to the membrane than the variant protein. In summary, we prioritize these top 10 genes on the left by filtering by mutational burden on exons, while also considering functional and structural effects within our scoring. AlphaFold modeling of structure, with its limitations in mind of the predicted mutations, found that most mutants, relative to their reference, were unlikely to be significant, while a few, including one for APOL4, were likely to have altered function, such as likely having a reduced binding affinity to membranes.